me just get my... Can you all hear me? Just out of curiosity. Blah de blah de blah de blah. Blah de blah. Say it, can you hear me? Okay, good. I don't know what's going on with Zoom. It's a pain in the posterior. Okay, now let's get everything up and running. Chat up. All right, it's really weird because before, when we first got Zoom, everything worked really, really well. And now that it's part of the university thing, it's just complete pain in the backside. Okay, so essentially what we're going to be doing in this section, so what we've done in the last section has been to give you a real solid foundation in kind of classical transmission genetics, like Mendelian genetics. And the reason for that, other than the fact that it's important to know, is that then allows you to look at the really interesting stuff, right? The cool stuff that actually explains much more of what really goes on in terms of genetics than the really kind of tightly prescribed genetics of Mendel, right? And so obviously you can't really get the benefit of all that cool stuff without knowing the basics first. So that was what the first section was really about. This section is all about everything else in terms of transmission genetics. So it's a really fun one. I actually really enjoy this section, probably way more than the first section, to be honest, simply because there's just more cool genetics involved. And you get to talk about history, like the, actually, I won't tell you who those are. Some of you might already know. Anyway, so this section really is all about kind of three different ways of looking at non-Mendelian inheritance, right? So basically, it's all about breaking Mendel. So the first thing that we're going to cover is sex determination and linkage, because they're pretty cool, right? And they're fun things to talk about. And also we get to talk about some really neat molecular mechanisms too, right? So I'm a molecular geneticist really at kind of my core. And so for me, I love talking about this stuff because it's really like, how does stuff work, right? It's all well and good knowing that this does this, does this, does this, that's fine. But what really gets me out of bed in the morning, I guess one could say, other than my alarm and the fact that I have to teach, is how does stuff work, right? Why does it do that? Why? It's like being a 3-year-old, right? Why? Anyway, sorry, getting a little bit exercise there. 2, what we call kind of non-Mendelian. And this is kind of like a catch-all term, really. It's a bunch of stuff, particularly about dominance, epistasis. Do we talk about epigenetics? I think that's later on. And we'll kind of cover what those things are, obviously, as the time comes. And then the third one, which is really interesting, right? And it's actually one thing that explains the inheritance of linked traits, right? And if you think about it, I'm always trying to get students to think about their own kind of genetic history in a sense, right? Because in many ways, you know, transmission genetics is it's like a historical thing as well as a forward-looking thing, right? You look back at your family tree as well as looking forward to what might occur in the next generation and so on. And if you think about how, your ancestors looked, or even, kind of parents, grandparents, things like that, you'll tend to notice that there's particular kind of traits that go together, right? One thing that I always find really curious is allergies. So I'm not rubbing my nose because of that, but things like asthma, hay fever, eczema, right? Kind of allergic reactions tend to go together, right? So if you have one, you most likely have another one as well, right? And those kind of travel down through generations, right? And so why do you get, obviously, because like, the mucosal surface of your nostrils, right, have, oh, and asthma. Did I say asthma? I can't remember. Maybe not. Right, the mucosal surface of your nostrils or your lungs are very, very different from the epithelial surface of the skin, right? So clearly there are different genes involved in those different kind of aberrant processes. So why do you get them all together? So really the one way of thinking about that is something called linkage. And this is a phenomenon. I think it's actually like mustache hairs are kind of sticking up my nose. I need to do what you do, Saeed, and kind of get them under control. It's a good idea. And so basically, linkage breaks the law of independent assortment. It's all about alleles traveling together, right? And so those are the three things really we're going to cover in detail in this section. And there'll be a bit of DNA replication at the end, and largely the reason for that is we need to talk about homologous recombination, which is kind of the actual mechanism that underlies linkage. Linkage is actually a bit of a tough topic to learn in many ways because there's a lot of math involved, right? And so, throw math into the mix and things get, a little bit harder. So essentially in all of that, and cripes, that's like 3 weeks. It's not very long. Takes us up to the next exam. Just never stops. Okay. So, a little bit of a history lesson, because this is fun. Does anybody know who this group are? Just out of curiosity. The ones in the picture there. Anybody recognize who they are? There's lots of interesting history kind of associated with that family and genetics as well. No, cool. Well, you get to learn something different. Yay, good stuff, Louise. It's the Romanovs. I think it's a V rather than F, but I'm not 100% sure. Yeah, they're the Romanovs. So these were the kind of the Russian royal family, essentially, at the turn of the century. And that chap with a very elegant mustache and beard. Yeah, handsome chap. Was Tsar Nicholas, right? So he was essentially the king of Russia when the Russian royal family were overthrown by the Bolsheviks in the October Revolution in 1917. Largely, and this is kind of a fun fact, facilitated by the Germans as a way of getting the Russians off their back in the First World War. So it's like, hang on, we're kind of struggling on both sides here, so let's go screw up the Russians, and then we'll just have to deal with the French and the English instead, or British. Anyway, so Tsar Nicholas and Alexandra, and they had 5 kids. And the reason why this picture here is that this family had a sex-linked disease in which this chap here, Alexei, was a hemophiliac. Right, and hemophilia is and we'll get into Yeah, we'll get into the mechanisms that are way later in the course, actually in the kind of the molecular stuff. So you'll learn the molecular mechanism of hemophilia, but we're more concerned with the genetics of it right now. Anyway, he was a hemophiliac, but he didn't actually, oh, and someone yesterday told me that Rasputin, everyone's heard of Rasputin, kind of like the dodgy geezer of the Russian court who supposedly liked the ladies a lot. He was said to have actually cured Alexei of his hemophilia somehow. Anyway, he never actually had, he didn't die of hemophilia because it's kind of what happened to most kids, boys that had hemophilia in that kind of era, because they were all assassinated by the Bolsheviks and buried in an unmarked grave in Siberia. And so there's actually a bit of a, I wouldn't say conspiracy theory because that's not quite the right term, but this long-running theory, because that grave wasn't found until very recently, that one of the daughters, Catherine, don't actually know which one of them it is, survived somehow and was, you know, tooling around Europe as a long-lost heir to the throne. Anyway, they finally found the grave in Siberia, and there were 7 skeletons of the appropriate sizes, and so that kind of put paid to that idea. So anyway, that's just a cool bit of history. I'm a bit of a history buff. I tend to kind of throw things like that in whenever I can, just because it's cool to talk about. So we'll get to that later. There's a real cool, where'd it go? There we go. There's a cool family tree there, which we'll kind of walk through in a bit, maybe today, hopefully today, if we get around to it. Anyway, so this lecture, and probably today, I'd say, is going to be all about sex determination and sex linkage or sex-linked inheritance, right? And so we're going to go through sex determination first, and then we'll finish up with some of the curiosities of sex linkage. And so essentially, this is talking about sex. This is all about dioecious species. And so dioecious species essentially are ones which reproduce sexually. And so that's the combination of haploid gametes in the next generation to create the next generation. And so there's lots of different ways in which one can determine sex: environmentally, genetically, chromosomally, even socially, right? If you think about, pardon me, the one-child policy in China, and also inheritance laws in India, which have largely led to a male bias sex ratio, right? I think it's about 55-45 male to female in China, which leads to all kinds of weird sociological things. Anyway, we won't get into that because that's a whole nother cup of tea. So probably the most common example, and this applies particularly for reptiles for some reason, is in terms of environmental sex determination, is temperature. And so it's a snapping turtle, I've been told, and another kind of turtle, whatever that might be, and an alligator. And the really neat one is particularly the alligator, right? Because essentially from like 32 to 35 degrees, you go from all female to all male to all female again, right? And so there's a very narrow window in which those eggs survive. Essentially, the eggs are buried in a kind of a pit covered by the mama alligator. And depending on how covered those eggs are, essentially determines the temperature. And the temperature determines the sex. I don't actually know the mechanism by which that happens. I'm not sure if it is known. I personally wouldn't want to work with alligators. They're big and scary creatures. Worms are much easier. And also, I can't keep alligators in the lab. I mean, can you imagine how many students I'd get through? Terrible. Anyway, So most likely it's something to do with temperature sensitive gene expression, right? So essentially proteins taking different shapes depending on the temperature and being either active or inactive as a result. But anyway, it's cool to kind of think about that. Now, the next kind of chunk that we're going to talk about is really sex determination, both in terms of what chromosomes you have, in terms of sex chromosomes, and also the molecular mechanism or mechanisms which underlie that, right? So first, you obviously need to go through the fact that we have two different types of chromosomes. We have sex chromosomes. They're the easiest to define in many ways because they basically differ by size, by shape, number of genes. Essentially, they differ somehow, and that difference is tied into the sex of that individual. There are lots of different kind of ways in which you can do that, which we'll get to in a little bit. Now, autosomes, that sentence isn't quite correct, as you'll find out later. Generally speaking, they don't play a, well, some of them do play a role in sex determination. They're not conventionally thought to have a role in sex determination. Now, obviously, that's a bit of an oversimplification because I'm going to show you how protein produced from an autosome does actually have a role in sex determination. So take that one with a, not a dose of skepticism, but a realization it's a pretty broad simplification. So really, that's probably more accurate. Another thing that's worth pointing out, and which you probably already know, is that essentially you have a difference typically between the sexes where one is typically heterogametic, in which it has two different sex chromosomes, and the other sex will be homogametic, which is where you have two of the same, right? So females in mammals, for example, are XX. That would be a homogametic sex. There's also, I keep changing this stuff. I'll upload a new version of this in a bit. Yeah, I'll just change this. There you go. I do like to be correct. Well, obviously, everyone wants to be correct. I do like to tell you the stuff that's correct. I don't want to kind of give you a gross simplification because I think we end up losing a lot of really interesting stuff as a result, even if some of those things may kind of sometimes fly over your head. And so there's actually, and this actually gets at how many different ways you can do this, right? So the ones that we're familiar with, like XX, XY, mammals, some insects, and birds like chickens are a little weirder. They have what's called ZZZW or ZZ, ZW. There are also some insects and nematodes, including one nematode that I did my PhD on, which is actually XXX nothing. So the males are X nothing. So male so sperm in these organisms do not have a sex chromosome. You only get sex chromosomes from, well, some sperm do not, some sperm do, but only when you have two sex chromosomes is it a female. So that's also a kind of different way of doing things. Oh, actually, another fun fact. I try and give you fun facts. Going back to environmental sex determination, the species that I worked on, which was a parasite of rats, it could change the sex of its offspring based on the immune response experienced from its host. So normally it produced females, which would then reproduce what's called parthenogenetically, which is kind of one way of reproducing asexually. But if it started getting attacked by the host's immune response, it would generate males as well. So those would reproduce basically in the rat poo. So that was a pretty cool mechanism to study. Okay. So what I really want to focus on now, and this will be a fairly solid chunk of the rest of the class, are two different mechanisms by which you can use XXXY to determine sex, right? And so one of the reasons to do this is because it's interesting, it's fun, and I get to talk molecular genetics, even though this is transmission genetics. And also it's a really good example of convergent evolution, right? Which is essentially where you end up with the same end result, XXXY sex determination, via very, very different evolutionary paths and different mechanisms. Right? So it's just a fun thing to talk about for that reason. So I'm going to wander over to the board. Let's see. Now a second. Yeah, that'll do. So what we're going to do is going to do sex determination in Drosophila first, right? And so the crazy thing about this, and this is a very weird concept to kind of get, is that in Drosophila, every single cell, right, every single one, and there are lots of them, determines its sex independently. So one hopes all the cells come to the same conclusion, but this is done at a cellular level, right? It's not done at the organism level, which is what happens in humans. And there's actually, I've been always do a bunch of reading on this stuff because I like to be able to understand what I'm teaching. There's so much more to it than what I'm telling you. This is actually a really good example of a developmental cascade, right? So one thing that leads to another, which leads to another, which leads to another. Anyway, we're not going to get into that right now because it's just a little bit too crazy complicated and I don't fully understand it myself. So essentially in Drosophila, we have XX are female and XY are male. The Y chromosome has nothing to do with sex determination in flies. So that's all involved in some sex-dependent traits, like sperm production, for example. Right? So once sex has been determined, then some of the genes on the Y chromosome have a role in maleness. But the Y chromosome is utterly irrelevant in terms of sex determination. That's obviously very different to humans. And so essentially what Drosophila do is they compare the number of X's to the number of autosomes. So with females, if you have a 1-to-1 ratio of X to A, you're female. If you have a 1-to-2 ratio of sex chromosomes to autosomes, you are male. So the big deal in Drosophila is it's a ratio. The Y chromosome has nothing to do with it. Everything in terms of sex determination in Drosophila is how many X chromosomes do you have? Do you have 2 to 2 autosomes, or do you have 1 to 2 autosomes? That's the deal. Anybody got any questions so far? Because we're going to get crazy. So essentially, there's one kind of key switch gene, right? So this is like a molecular switch where if you switch it on, you'll get one particular outcome. If you don't switch it on, you'll get a different outcome, right? And so it's a very binary deal, one way or another. Anyway, sorry, don't want to start dancing. And this gene is called sex lethal. The reason being is if you knock it out, you get dead flies. Right, so no functioning sex lethal equals no functioning fly, essentially. So in females, and I'll get to, actually, let's put this a little bit further down so I have space. Essentially, if sex lethal is on, it promotes femaleness. And it inhibits maleness. And that kind of like blunt arrow, inverted T shape, is a genetic symbol for inhibits or suppresses. Right, so this is if the switch is switched on, you get females, right? And again, remember, this happens at the level of the cell. If sex lethal is off, maleness is allowed to happen, I guess. It's kind of not quite the right word. But in the absence of sex lethal or being on, functional, essentially expressed, right? That's how it works. You get male cells. Right, so that's the switch. And actually it's pretty crazy. Trying to get the reflection in the right place. Bloody lights. Can you read that okay? Cool. The actual mechanism by the way this switch works is really crazy. It's actually an RNA binding protein, right? So it changes how other genes, how other mRNAs are spliced. Yeah, I told you it's crazy. I actually read through a bunch for yes, I was like, okay, I'm going to do something which requires less brain power now. So how do you turn these off, right, and on? How do you count this ratio between male and, you're welcome, Louise, male, well, X chromosomes and autosomes? And essentially what we deal with here is something that's produced on the X chromosome and something that's produced on the autosome. So now we're talking about gene products. And essentially, the more autosomes you have, the more gene products you have relative to how much is produced from the autosomes, more sex chromosomes, I mean. And so the X chromosome has a gene which is called cystalis. Now, I'm sorry, this is just Drosophila people. They love calling genes silly names. They all have a purpose, sort of, but they're still bloody irritating, in my opinion. And cystalis, because if you don't have cystalis, you don't have sisters. Anyway, it's much more logical and sensible with worms. Trust me. Actually, we used to have, this is a bit of a tangent, one guy, a guy called John Hopkins, who works at Cambridge, University of Cambridge in the UK, and he vetted all gene names from the entire worldwide worm community. I was like, yes, no, that's silly. And so he was like the global curator of gene names, just to prevent this kind of nonsense from happening. There's actually a gene in Drosophila called shaven baby, for example. How ridiculous is that? So anyway, cystalis is an inhibitor of, or is inhibited by, so it turns on deadpan, actually, hang on, let me get this right. It inhibits a gene called deadpan. I don't know why it's called deadpan. I always think of Deadpool for some reason whenever I teach this. And deadpan, so this is where stuff gets crazy. I could kind of warn you, right? So for me, I love this stuff. Other people are like, what the hell is he talking about? The role of deadpan is to inhibit sex lethal. So essentially what you have is a balance between these two things. If you have equal amounts of cystalis and deadpan, cystalis stops deadpan from inhibiting sex lethal. And so now we're essentially counting chromosomes, because if you have two X chromosomes producing cystalis and two autosomes producing deadpan, you have the same amount. They essentially cancel each other out. It's like 1 minus 1 or 2 minus 2, right? You end up with nothing. And if you don't have any of this jazz going on, sex lethal is on. Does that make sense? So that's the counting mechanism. You're actually counting the relative amounts of two proteins.
Did you say that if you have equal parts cystalis and deadpan, the sex lethal is on?
Correct.
Okay.
Because the role of deadpan is to turn sex lethal off. And so if there isn't any deadpan around because it's been canceled out by cystalis, essentially this stuff all disappears. Doesn't disappear, it's still there, but functionally it isn't involved in anything. So sex lethal is left to be, on its own devices, essentially. Does that make sense? Good question, Jesus. So that's a hard question to answer. So what are the evolutionary benefits to doing sex determination this way? To be honest, you'd have to look at the evolutionary history of the animal and the different genes involved. So it's hard to think in terms of benefits because I don't have the context, if you see what I mean. So I couldn't actually answer that question. For a different system, for C. elegans, yes, I could answer that question, but that's a far more complicated process, which I don't fully understand. So it's the cheap and easy answer is essentially there's a bunch of different genes involved in something, at some point in the evolutionary history of Drosophila as a genus, and they accumulated these roles, and this is what you get, essentially. Also, Drosophila is a very weird organism in that it, in development, it doesn't actually have individual cells until fairly late in relatively speaking, in development. It has a bunch of nuclei which have a shared cytoplasm. So that may be part of why it's done at a cellular level rather than an organismal level like it is in humans. That's kind of my best guess, to be honest. I don't actually know. So if you have equal cystolus and deadpan, you'll have females.
And so then if you had unequal, would you have more male-less present?
Correct.
Okay.
That is exactly right. So if you go over here, right, now we have fewer X chromosomes. We only have one. Well, yeah, kind of. Two autosomes. So now we have, we're still producing cystolus, but we're just producing like half the amount of it relative to what we have in females. We still produce the same amount of deadpan because the number of autosomes does not change, only the number of sex chromosomes or X chromosomes. So now we don't have enough cystolus to inhibit deadpan, and deadpan turns off sex lethal. And with sex lethal off, now you have males. See, I told you it was crazy. I wasn't lying. Did that help? Does that make sense?
Yeah, thank you so much.
Yeah, it's not a trivial process to get by any means. I'm kind of pretty aware of that. But the reason why I want to go into this in this kind of detail is because it's a really cool mechanism. It's very elegant, really, if you think about it. Being able to actually count chromosomes by the amount of what they produce, in terms of gene expression. And it's also kind of a first, if somewhat brutal, introduction to the concept of genetic pathways. So different genes interacting to give in different ways to give two different outcomes. And also, it's just a bloody hard thing to explain if you don't go into this kind of detail. Does anybody else have questions before we move on? I will take a picture of this and post it to Blackboard, so don't stress if you don't have this all down or you can't read different bits of it. Cool. If you do have questions, just pipe up and ask, because, you know, obviously, ooh, that's the point. So, cool thing is, humans and mammals also have an XXXY sex determination mechanism. However, and also there's like lots of details down here as well, if you need to kind of look at that more. So humans instead do this at a whole organism level. And so I'm not much of a human embryologist, to be honest. I kind of, in terms of my interest in human development, kind of stops fairly, no, yeah, it certainly stops by birth. So anyway, in human development, at 6 weeks post-fertilization, which is actually kind of hard to tell when that is, but 6 weeks post-fertilization, the embryo, which is basically like a little grain of rice, well, cashew nut, I guess. The embryo has both proto-male and proto-female reproductive tissues. It is essentially uncommitted. Right. And so the proto-female tissues are called the Mullerian ducts. That's not quack. That's like what carries cold air around your house. And the proto-male tissues are called Wolffian ducts. So essentially, these are tissues that have the potential in later on in development to develop into the reproductive tissues, or at least most of them. So in females, the Mullerian ducts develop into the ovaries, fallopian tubes, uterus, and upper vagina. In males, the Wolffian ducts instead develop into the testes, epididymis, and some other bits and pieces. Can't remember all the different names of them. Right, so not all of it, but most of it, essentially. But at 6 weeks, you actually have both of those tissues present. They kind of lie sort of parallel to each other, just above what will become part of the urinary tract, essentially. Development is just such a funky thing to study. It really is. It's an astonishing subject. Anyway, so at around 6 weeks of development, post-fertilization, a switch occurs. Now, this is even though, you know, the embryo is pretty minuscule at that point, this switch occurs over the whole of the embryo, right? So this isn't a cell-by-cell deal like you have in Drosophila, where actually if it goes wrong in one cell, you can actually technically have male and female cells inside a male or a female fly, which is really weird to think about. It's kind of whole organism process for mammals. And so this switch occurs, and I'm not actually sure what switches that switch, right? Because something must, right? And it must be involved in what sex chromosomes you have in some way or other. But there's a gene on the Y chromosome called the sex determining region Y, or SRY. And so actually, if I'm going to be There you go. Sorry, being a pedant again. Lowercase in italics is a gene, uppercase in capitals is protein. So only males have this gene, right? So if you're a genetically female, so you're XX, you do not have this gene, right? This is one of the rare occurrences where there are genes that are present in one sex and not another. So in the embryos that have a Y chromosome, you essentially turn on this gene called SRY, and this is the switch that results in the cascade that ends up with male and female embryos, male or female embryos. And so it is a transcription factor, very different from sex lethal. And it turns on a whole bunch of genes, right? This is called a expression program or a developmental program. So this protein, and we'll get into transcription factors in more detail a lot later, this SRY protein recognizes the control regions of a bunch of different genes involved in male development. And so when those genes get expressed, again, this is a kind of a developmental cascade in a sense, the testes start developing, right? So the Wolffian duct starts developing into a separate array of tissues, one of which includes the testes. Now, these testes produce two different things. One is a hormone, which you're all familiar with most likely, testosterone. Right, so it's commonly called a sex hormone, but really it's involved in a lot more than just sex determination. It's a bit of a misnomer, to be honest. So this is a compound, it's a sterol, it's like a type of a fat, basically. And it's very, very, very similar to estrogen, estradiol, and what's the other one? I can't remember. Anyway, they're very, very similar to each other. And testosterone basically promotes kind of maleness, right? So over the course of development, higher bone density, higher muscle mass, potentially, various other secondary sexual characteristics and the like. However, don't forget there are two potential reproductive tissues in that embryo. And so the other thing that the testes produce is something called the Müllerian inhibiting substance factor. It's got a lot of different names, not all of which I can remember. And its job is to cause the Müllerian ducts to degenerate. And so not only do you have pro-maleness, but you have anti-femaleness. And so those ducts those Müllerian ducts just disappear, essentially. They get reabsorbed into the surrounding tissues. Now, if you don't have a Y chromosome, because you have two X's, SRY never gets turned on because it's not there. And so all those other genes, many of which are on your autosomes, both male and female, those other genes involved in testes development don't get turned on, right? So those Wolffian ducts in female embryos, or what will become genetically female embryos, that never gets turned into testes and other stuff, right? So that in essentially the absence of a pro signal, like do this, the Wolffian ducts degenerate, and so you're left with the Müllerian ducts, which develop into the female reproductive tissues. So you can see, even though Oh, that's an interesting question, Michael. Give me just a second. So even though you end up with the same thing, male and female sexes as a result of XXXY, the way in which you end up with that is very, very different. So hermaphroditism, essentially, yes. So there's different aspects of that. If I'm just gathering my thoughts here. If you have aneuploidy of the sex chromosomes, you can have XXY, which is Klinefelter's, and essentially you have SRY because you have a Y chromosome, but you also have too many X's, and so you end up with kind of a sterile intermediate, essentially, right? So individuals with Klinefelter's are sterile, but kind of male, but also with, you know, female characteristics as well. If you're X nothing in humans, you're female, but you have a lot of other issues, right? And end up essentially sterile, almost certainly. So if you mess around with expression of SRY, then you can end up with both sets of reproductive tissues. How that's expressed in terms of gross physiology depends on exactly how that expression is messed up. But yeah, you can certainly end up with, and to be honest, I've read about it a bunch, but I don't understand it well enough to explain it very well. But you can end up with both or some aspects of both tissues developing into reproductive tissues. How functional they are very much varies. So you can have externally males, or, you know, in terms of having male reproductive organs like penis and testicles and stuff, but they'll also have a uterus and ovaries, but those won't be functional, or the penis and testicles won't be functional, and vice versa. So actually it's kind of more on the sort of male and stuff side rather than the female and stuff side, because you don't have SRY if you're a female. So yeah, if you mess up this process, you do end up with kind of partial development of different tissues, right? Those Müllerian ducts do not fully degenerate, right? And so if they're still there, then they can receive signals from other stuff and develop into other things. So yeah, it's complex. And to be honest, I don't really kind of have a good enough grasp on it to explain it very well. But if you go up and do some reading on your own, you'll find a whole bunch of stuff about this, which is really interesting to read. Essentially why I did. So yeah, sorry I don't have a very good answer for that, Michael, but that's but it is an interesting question. Any other questions about sex determination before we move on to sex linkage? Okay. So now, essentially, because you have different numbers of X chromosomes or presence of a Y as well as an X, depending on where genes are in terms of X versus Y, you can get traits which are inherited in a sex-linked manner, right? And so these are typically, because there aren't actually that many genes on the Y chromosome, it's fairly like tiny, sparse little nubbin of a chromosome. Most of these genes we're talking about are on the X chromosome, but, and there's a big, big but. Sorry, that's kind of infantile. If you only have one X chromosome, you are what's called hemizygous. And what that means is whatever allele you have for a gene on the X chromosome, that will be expressed. So this is very different from autosomes and standard Mendelian genetics, and that's worth, writing down. So because for autosomes, the phenotype you express is dependent on whether or not you have at least one dominant allele out of two. On genes on the X chromosome, if you're female, that also applies, right? Because you have two X chromosomes. If you're male, you only have one X. So if you only have one X, whatever you have for that gene is what you get. Can anybody think of some X-linked diseases in humans? We covered one, or well, introduced one already, right at the very beginning.
The hemophilia?
Yeah, hemophilia is one. The blood clotting disorder, which can lead to you literally bleeding out from a scratch. What else? Think of other things which afflict guys, basically. Now, I actually don't have any of this in my family. My uncle was, but no one else is. So I have a full head of hair, and hopefully will continue to do so. What else is you tend to see in fairly commonly in males? Yeah, color blindness most definitely counts. So color blindness is another classic example as well. So it's very, very rare to find a woman that's red-green colorblind. It's fairly common in males. That's another good example. It's actually spawned entire industries of hair dyes and transplants and all kinds of stuff. Big business. Someone must have a relative who is this. Which I don't know. Yeah, male pattern baldness. That's the other one. Actually, this is just a kind of question to the class, right? Because many of you have Hispanic background. Do you tend to have lots of people that are bald in, Hispanic populations? Because I'm not, and I kind of grew up in a Northern European type population. Do you know many people that are bald? Like fathers, uncles, grandparents, stuff like that? Okay. Yeah, it's just interesting because just like thinking about all the people that I've interacted with over the years since moving to San Antonio, I don't seem to remember all that many people that are bald. Yeah, anyway, just out of curiosity, really. Certainly not enough to make any kind of statement about it, but it's just something to think about, just something interesting. Anyway, maybe, get too much sunburn, not an issue in Northern Europe. So those are all things which you'll see fairly commonly, obviously depending on background, in males, but you will very, very rarely see them in females. However, females can carry the alleles for those, right? So males will express the phenotype, baldness, etc. But females will carry or can carry recessive allele. And so they're called carriers because they're heterozygous. Let's see what I've got. So this is something that was really studied early on in the advent of genetics. Thomas Hunt Morgan was the kind of pioneer. He was really the father of genetics, to be honest, kind of like modern era genetics. And actually, you'll be working with these this trait in lab, which is kind of neat as well. So the white gene, which gives flies with white eyes, like the ones you saw in the last lab, is a sex-linked or an X-linked trait. And so even though we're not looking at sex linkage in the lab, these genes are on the sex chromosome. And so, pardon me, one classic characteristic of sex linkage experimentally, right, not talking about lineages, but experimentally, the classic characteristic is you'll get different results depending on which way round you do the cross. And so if you remember way back when we were talking about Mendelian genetics, this is called a reciprocal cross, right? So if you have a male recessive and female dominant, you get 3-to-1 ratio. If you put it around the other way around, you'll still get a 3-to-1 ratio, right? It doesn't actually matter. But for sex linkage or sex determined traits, right, it does matter. So if the male is recessive, right, because it only has one copy of that recessive chromosome, essentially, recessive allele, you'll end up with the classic 3-to-1 ratio, right? Just as you would in a normal monohybrid cross. Now, if you do it the other way around, now you have the recessive female, right? And actually, it's funny because this first part of the cross here, these two boxes on the left in the left column, that's exactly what some of you will be doing today, right? So this is the first cross that we will do for our fly linkage project, which is kind of neat. But if you do it the other way around, now you have two chromosomes. So you have two recessive alleles kind of in the mix, essentially. And so the F2 generation will actually give you a 1-to-1 ratio or 2-to-2, right? So reciprocal crosses give different results depending on which is male and which is female in terms of the alleles, right? So that's a classic characteristic of sex-linked inheritance. The other one, let's move this out of the way so I can see what I'm pointing at. The other one is that you only see it in males, right? I mean, it's kind of self-evident almost, right? So this is the European royal family, Queen Victoria here and Albert. It's actually a museum. It's a really cool museum if you ever get to travel to UK, in London, called the Victoria and Albert Museum, full of really, really cool stuff. Anyway, Queen Victoria was a carrier. So she was heterozygous for the hemophilia allele, right? And she obviously had lots and lots of descendants. Show you something really disturbing. First off, which may explain some of what Prince Charles is the way Prince Charles is. Don't know if you know of him, but he's a bit of a weirdo. If you look down here, here's Prince Charles. Should actually put like a big pair of jug ears on it just to kind of make it obvious. But his mother and father, Queen Elizabeth, yay, love Queen Elizabeth, and Prince Philip, rest in peace, essentially had the same great-great-grandparents. So Philip came all the way this way, and Elizabeth came all the way this way, and basically they had the same relatives. Isn't that disturbing? Just can't be good, right? Thankfully, the offspring of Prince Charles has done a little bit of outbreeding. And so even going to the US, of all things, who would think of it? And so there's a whole bunch of fresh alleles coming into the royal family, which is a good thing. Anyway, so if you look at this, right, so we have 2 of Queen Victoria's offspring were also carriers, Alice and Beatrice, right? And so you will see in their offspring hemophiliacs in males. But because this is a recessive trait, and until, recent times, hemophiliac males didn't live long enough to pass it on to their offspring, because hemophilia, well, I don't actually have ages here. There are different genealogies which have the ages in there. They typically die by their early teens, simply because it's just a matter of time before they have a cut that's big enough that it will not stop bleeding. It's actually a pretty horrible way to die if you think about it. And so if you look through this whole lineage, squares are males, round are females, full color is the disease phenotype. You'll only ever see males having the disease. And so if you have a male that has the disease, that male has to have gotten that recessive allele from his mother, because it's on the X chromosome. And so you can actually, Leopold's actually seems to be the exception here. He managed to make it to reproductive age. But you can follow this one all the way through another Alice, who is a carrier, through Leopold, who was a hemophiliac but lived long enough to have offspring, all the way back to Victoria. In other lines, it disappears, right? So if just by chance, right, because it is random, like how many offspring you'll have that are carriers versus non-carriers if they're female. As you can see, Victoria had one, bloody hell, she was busy. 1, 2, 3, 4, 5, 6, 7, 8, 9 offspring. Wow, just tires me out thinking about it, right? Of those 1, 2, 3, 4, 5, yeah, 5 females, you'd expect about half of them to be carriers, and there are 2, right? Obviously, it doesn't round easily. So, and you'd expect about half of the males to be hemophiliac, right? But it's actually a little bit lower than that. It's 1 in 4 instead, for some reason. Don't actually know why, just by chance. And so you'll actually see that disappear, right, over time. It's fairly rare that hemophilia carries on for a long time if outbreeding occurs, right? Obviously, if you're still dipping from the same genetic pool, no guarantee whatsoever. But yeah, thought that was fun. Also, and just to quickly wrap it up, the Y chromosome is pretty weird, right? It's very small. Only males have it, right? So there are genes on there which females do not have because they are present only on the Y. And because it's so small, right? So this is actually a kind of an oddity if you think about it, right? During meiosis in males, we have the homologous chromosomes have to pair up, right? And they pair up to recombine and hold together until metaphase 1, right? You can't have recombination between Y chromosomes and X chromosomes, right? They're different sizes. They have different genes on them. It would be catastrophic, right? So there are actual regions at the end which kind of stick together, right, to hold them together as if they had recombined. But there is actually very little recombination because there's nothing to recombine with. There is recombination between X chromosomes if you have an X and an X, but not in Y chromosomes. And so the cool thing about that is essentially Y chromosomes will have a set of alleles which doesn't get mixed up, right, through recombination. On your other chromosomes, your autosomes, every time you produce gametes, those alleles will be shuffled to one greater or lesser extent. Doesn't happen in your Y chromosome. And so now we have what's called a haplotype, haplo meaning half, of your Y chromosome, where we have a list of alleles that tends to stay the same, right? The only way that that can change is through mutation, right? And so we can actually use that to look at inheritance and descendants, right? And so cool example of this is the Mongol Empire, right? From Genghis Khan and Kublai Khan. And that is actually another bit of history. One of the most amazing empires in history, right? They were able to control somehow an empire which spanned from the Pacific, right? The Pacific coast of Russia and China, all the way to the Stans, almost to the Black Sea, almost to Europe, right? If you think how big of a landmass that is. And they're actually the first, every time you write a check, which most people don't anymore, but checks were actually invented by the Mongols so that they could carry money essentially in paper form all the way from one part of the empire to the other without actually having to hoof it around. Anyway, if you look at the Y haplotypes of individuals, of males from this region, you'll tend to find haplotypes similar to what presumably came from Genghis Khan, because he was, I imagine, a prolific chap as well, which maps essentially to the ancient kind of boundaries of that empire. So you can actually look at how populations move, right? How they move around, how they breed, interbreed, things like that. And one of the curiosities are, and these have actually been in the news quite a bit from all the tumult in Afghanistan, but the Hazaras, which are, I think they're a Muslim sect, I can't remember off the top of my head, have a very high instance of that haplotype. So this is a distinct population, which although outside of the ancient boundaries of that kingdom, has stayed together after migrating to present day Afghanistan and northwestern Pakistan. So you can look at evolutionary and migratory and population histories literally over, well, that's probably about 10,000 years would be my guess. The Khans were around, I think they're around 4,000. Oh, actually, I can't remember. No, I can't remember when I think that was in the ADs. Yeah. Anyway, I'll leave it there before I tie myself in knots. But it's a really, really interesting way of thinking about it. It's kind of, as with mitochondrial DNA, you can trace lineages down the matrilineal line. With Y chromosome haplotypes, you can do it down the patrilineal line, which is really, really cool. Anyway, that's enough from me. Well, some of us have lab in just under 15 minutes. So I'm going to sign off and stuff some food down my neck. And I'll either see you in a little bit, or I'll see you on Wednesday or Thursday. All right. Have fun, everybody.